Auzu billahi minash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Dear student, Assalamu alaikum. Today is our 28th lecture on rational drug design. Today we will discuss about the structure-based drug design. The today title of the lecture is structure-based drug design. Key concepts, if we look at the key concepts, then drug design, a rational approach, introduction to drug and drug design, structure-based drug design. These are the key concepts today we will discuss in this lecture. This is very old style slides about the rational drug design. Rational drug design is also sometimes referred as drug design or rational design. It is a process in which finding of new medication based on knowledge of biochemical target. It involves design of small molecules that are complementary in shape and charge to biomolecular targets. The drug is most commonly an organic small molecule that activates or inhibits the function of the biomolecules such as proteins, which in turn results in therapeutic benefits to the patient. In contrast to traditional methods of drug discovery, which rely on trial and error testing of chemical substances on culture cells or the animals and matching the apparent effects to treatment, rational drug design begin with hypothesis that population of the specific biological targets may have therapeutic value. The meaning of rational drug design is that we are going to discover some specific molecule to treat the specific disease. You have to understand that the drug rational means most specific, targeted one. If we are going to discover for anti-cancer, the drug must be used for anti-cancer. It will be never used for other disease. This is the rational. To know the rational, it is very difficult method of drug discovery because usually we adopt hit and trial method. We adopt the random screening of the compounds from the library of compounds. Sometimes we synthesize, sometimes we isolate, sometimes we do microbial transformation, sometimes we isolate from the algae, sometimes we isolate the drug from the marine organism. A lot of methods are available for the discovery of new biomolecules. Usually biomolecules are the protein and carbohydrates, but proteins are involved in the therapeutic benefits for the patients because a lot of diseases are due to the destruction of the protein. When the protein is destructed, when the protein is translated, this is called post-translational modification of protein. Usually post-translational modification of protein causes the disease. This is the rational design. If we go towards, if I discuss introduction to drug and drug design, the drug is most commonly an organic small molecule that activate or inhibit the function of biomolecules such as protein, which in turn results in therapeutic benefit to the patient. Means drug is the most commonly an organic small molecules. Usually just see the example of aspirin, see the example of paracetamol. These are very, very small molecules, just benzene ring, and there is some OH group and some amide acetamide group on this make the paracetamol. Usually drugs are very small molecules. Sometimes big molecules, macromolecules are also act as drug, but usually organic small molecules that activate or inhibit. Activate or inhibits, the meaning of activate or inhibit is that means sometimes our system is suppressed. There is need to activate the system. Sometimes system is overexpressed. We need to inhibit the function. This is called activates or inhibits the function of the biomolecules. Biomolecules are protein. In our body, proteins are present. A lot of proteins in the form of enzymes. Enzymes are protein nature. Thousands of enzymes are present in the body. They are interlinked. They are interconnected with each other. That is why A lot of diseases are caused by the proteins. All the diseases which are caused, usually the basic cause of disease is protein. Proteins are causing diseases. This is the basic. The drug is the most commonly an organic small molecule that activate or inhibit the function of a biomolecule such as a protein, which in turn results in therapeutic benefits to the patient. Drug design or rational drug design, or simply rational design, is the inventive process of finding new medication based on the knowledge of biological target. Drug design or rational drug design or simple rational design is the inventive process. Invention means you are going to discover new chemical entity, new chemical compounds which has therapeutic value, which is used to treat the diseases. Is the inventive process of finding new medication based on the knowledge of biological target. Drug design involves the design of small molecules that are complementary in shape. Drug design involves the design of small molecules that are complementary in shape and charged to the big molecular targets with which they interact and therefore will bind to it. Are you listening to me, students? Please, you have to give me a response that you are listening. You have opened the slides of introduction to drug design, drug and drug design. Are you on that slides? Okay. Drug design involves the design of small molecules that are complementary in shape and charge to the biomolecular targets with which they interact and therefore will bind to it. Usually, drug molecules bind with the target. Which is target? Protein is the target. How molecule will interact? Molecule will interact through covalent interaction, through hydrogen bonding, through van der Waals forces. A lot of secondary interactions are present in the molecules. The molecules interact with the protein. Protein also abundant. Protein is abundant with the atoms like carbon, nitrogen, oxygen. Similarly, sometimes our drug molecule also contains carbon, hydrogen, carbonyl group. That is why secondary interactions are developed. Secondary interactions are developed between the target and the drug molecule. This is the drug design involved the design of small molecule that are complementary in shape and charged to the biomolecular target with which they interact, therefore will bind to it. The binding of drug to the target is due to the interactions. Which interaction? Which are the hydrogen bonding, van der Waals interactions, covalent interactions, pi stacking interactions. These interactions are involved which bind with the target. Then you can see in this slides, in the right side the picture, you can see the ribbon shaped structure is the protein structure. The ribbon shaped structure is the protein structure. Protein structure here you can see this is the ribbon type, and the ball and stick model of the compound is going to interact with the active site of the target molecules. Drug design frequently but not necessarily relies on computer modeling techniques. Nowadays, because computer modeling is the new technique, this is the emerging field of drug design. Drug design frequently but not necessarily relies on computer modeling techniques. This type of modeling is often referred to as computer aided drug design, CADD, computer aided drug design. This type of modeling is often referred to as computer aided drug design, in which you can see that the ribbon blue in blue color and in a green color and in red color and in yellow color and purple color. These are the ribbon shaped structure of the protein. This is the target molecules. This is the target molecules. This is the target. Target means this is the protein which is causing disease. This is our molecule which is you can see the ball and stick model, which is going to interact with the active site through different sometimes hydrogen bonding, sometimes it interact with the covalent interactions. Finally, drug design that relies on the knowledge of the three-dimensional structure of the biomolecular target is known as structure-based drug design. The phrase drug design is to some extent a misnomer. The phrase drug design is sometimes, this is the term which is used for the discovery of drug design. This is the misnomer. A more accurate term is ligand design. Ligand means that design of a small molecule that will bind tightly to its target. Means we are going to synthesize a small molecule. This is the ligand. A more accurate term is ligand design, that design of small molecule that will bind tightly to its target. Background of the structure based drug design is that biomolecular target, as for example proteins, nucleic acids, is a key molecule involved in a particular metabolic or signaling pathway that is leading to a specific disease condition or pathology, or to the infectivity or survival of the microbial pathogen. In some cases, small molecule will be designed to inhibit the target functions in the specific pathway disease state. Small molecules inhibitors or modulators. Small molecules can be inhibitors, they can be they can be modulators, will be designed that are complementary to the active site, allosteric site of the target. Usually the target contains two sites. One is the active site, which contains prosthetic group. Active site may contain a metal like zinc, like magnesium, like nickel. These are the active sites of some enzymes. The other sites other than active sites are called allosteric sites. Allosteric site, this is called allosterism. Allosterism is very common in drug discovery. Sometimes compound is going to bind to the other side rather than active site. This is called allosteric site of the target. In some other cases, small molecule will be designed or developed to enhance the normal pathway by promoting specific biomolecular molecules in the normal pathway that may have been affected in the disease state. This is the background that why there is need to develop new molecules, why there is need to discover new molecules, why there is need to invent new molecules. Usually diseases are increasing day by day. You know better nowadays, this the disease COVID-19. This is the viral disease, bacterial disease. All the diseases are caused due to the structural modification of proteins or nucleic acid. This is very common concept. Once again, I will tell you that the biomolecular targets like proteins, nucleic acid is a key molecule involved in a particular metabolic or signaling pathway that is leading to a specific disease condition or pathology, or to the infectivity or the survival of the microbial pathogens. In some cases, sometimes small molecules are designed to inhibit the target function in a specific pathway, because it is very important. If you are going to inhibit means you are going to reduce, not to finish. When you will finish the pathway, then it is also dangerous. Inhibit means reduction, because sometimes molecule cause overexpression, like target molecule proteins are overexpressed, overactive. There is need to reduce the overactivity of the target molecules, then we need inhibitors. That is why this is the biomolecular targets, small molecules, inhibitors or modulators. Modulators are those species which are going to modulate, means they regulate the pathway. If there is some irregularity, let's see examples that there is a metabolic disorder of sugar. Means if a patient is diabetic, why he is diabetic, he or she is diabetic. The basic reason is that the glucose level is high. Why glucose is high? Because there is some problem with the metabolic pathway. We give medicine in diabetes. A lot of enzymes are now like alpha glucosidase, beta glucuronidase, alpha amylase, DPP4. A lot of enzymes are involved which are causing diabetes. Inhibitors modulate the path of these enzymes. Sometimes in the market you will see there are some alpha glucosidase inhibitors, there are some beta glucuronidase inhibitors, there are some alpha amylase inhibitors. These inhibitors play an important role to reduce the sugar level in the blood. Otherwise, sugar causes many diseases. Sugar is a state, a condition of the body. If you will not control the diabetes, then it will affect your eyes, it will affect your kidneys. That is why it is very important to regulate, to modulate the pathway. In some other cases, small molecules will be designed or developed to enhance the normal pathway by promoting specific biomolecular molecules in the normal pathway that may have been affected in the disease state. Next, small molecules drugs can be designed so as not to affect any other important off target molecules or the anti-targets. Here you have to understand the point which I have already explained, that drugs should be specific. When there is a lung cancer, it should be only used to treat the lung cancer, and it should not affect your kidney, it should not affect your liver. That is why structure based and rational drug design is very important. Small molecule drug can be designed so as not to affect any other important off target molecules or anti target, since drug interactions with off target molecule may lead to undesirable side effect. In this point you can understand that since drug interaction with off target, on target and off target, this is the two concept. On target means the protein which is causing disease, off target means the protein which is silenced, which is not causing disease, but your molecule is going to interact with that silenced target molecule. This is called off target. With after molecule may lead to undesirable side effect. Sequence homology is often used to identify such risks. This is the technique through which we can understand that this protein is silenced and this protein is causing disease. This is called sequence homology, is often used to identify such risks. Sorry. Most commonly, drugs are organic small molecule produced through chemical synthesis, as I have discussed in earlier slides, that most commonly drugs are organic molecule, small molecule produced through chemical synthesis, but biopolymer based drug, also known as biologics, produced through biological process are becoming increasingly more common. Means आवाज आ रही है? Can you listen me? Okay. Most commonly drugs are organic small molecule produced through chemical synthesis, but biopolymer based drug, also known as biologics, produced through biological process are becoming increasingly more common. Means nowadays the biological targets, biologics produced through biological process, nowadays people are going to discover drug through biological process. This is the very common route to discover new drug molecules. Slides. Introduction to SBD. SBD stand for structure based drug design. S stand for structure, B stand for based, D for drug, D for design. Structure based or direct drug design. This can be also, we say that direct drug design. Structure based drug design or direct drug design relies on knowledge of the three-dimensional structure of the biological target obtained through methods such as X-ray crystallographic or NMR spectroscopy. We are using nowadays different methods to know the biological targets. As I have told you in my earlier lectures that when you need to discover drug molecules, then it is very important to discover, to know the target. Target, it is just like a person who is uh, doing some firing on the target. Means if a person has no target, then where he will do the fire. That is why when you are going to discover drug molecules, you must know the target. If you don't know the target, then you will not be able to discover new drug molecule. Structure based drug design or direct drug design relies on knowledge of three-dimensional structure of the biological target. If three-dimensional structure of the biological target is very important. Three-dimensional structures of a biological target can be only known with the help of techniques like X-ray crystallography or NMR spectroscopy, nuclear magnetic resonance spectroscopy. These are the techniques which are used to know the structure of the target. If an experimental structure of a target is not available, if an experimental structure of a target is not available, it may be possible to create a homology model of the target based on the experimental structure of the related proteins. Means sorry, if an experimental structure of a target not available, sometimes experimental structure of the target is not available, it may be possible to create homology model. Homology modeling is a technique through molecular modernity, through molecular mechanics. We do homology modeling. It may be possible to create homology model of the target based on the experimental structure of the related protein using the structure of biological target candidates. Drugs that are predicted to bind with high affinity and selectivity to the target may be designed using interactive graphics and the intuition of the medicinal chemist. Using the structure of the biological target candidate, drugs that are predicted to bind with high affinity and selectivity to the target may be designed using interactive graphics and the intuition of medicinal chemist. Sometimes medicinal chemist initiate some process to know the interaction of the molecules of drug candidates which are going to interact with the target. Structure based drug design, I think this is the same slides which I have told you. Next slide play, you can see that this is the X-ray, simple X-ray crystallographic techniques in these slides. This is the X-ray beam, this is the diffraction rate, these are the crystallized molecules, this is the film. How crystallography helps to determine the three-dimensional structure of the molecules. This is NMR. In this slide, you can see this is the structure of NMR. The instrument is very big. You can see that these are stairs which are used here. These stairs are used by the technician. He go up by using these stairs, and on the top he put the sample in the probe of in the NMR. Then he start to know the structure, 3D structure of the target. This is the homology modeling. Homology modeling, this is, you can see that these are the different ribbon like structures: purple, blue color, red color, yellow color and green color. These are the these are the homology modeling. These homology modeling are used to determine the structure of target molecules. Because it is very clear to you, you must understand that if you don't know the structure of target, then it is very difficult to discover new chemical entity. Structure based drug design, in next slide you can see that introduction to structure based drug design. Structure based drug design is one of the first techniques to be used in drug design. Structure based drug design is the first technique which is used in the discovery of new drug molecule. Structure based drug design that has helped in the discovery process of new drugs. In parallel, information about the structural dynamics and electronics properties about ligands are obtained from the calculations. We do some calculation, some chart density of the ligands, some electronic properties. In parallel, information about structural dynamics and electronic properties about ligands are obtained from the calculations. Calculations are usually through through quantum mechanics, through density function theory. This has encouraged the rapid development of structure based drug design. This has encouraged the rapid development of the structure based drug design. Structure based drug design can be divided roughly into two categories. Usually we roughly divide the structure based drug design into two categories. One is ligand based drug design or database searching, and second is receptor based drug design. These are the two major categories which are used in the structure based drug design. One is ligand based drug design or database searching, and second is receptor based drug design. Structure based drug design can be divided roughly into two categories. You must understand there are two categories of structure based drug design. One is ligand based drug design or database searching, and other is a receptor based drug design, receptor based drug testing. These are two categories which are used in structure based drug design. Ligand based drug design, the first category is about finding ligands for a given receptor. The first category is about finding ligands for a given receptors, which is usually referred as database searching. In this case, a large number of potential ligand molecules are screened to find those fitting the binding pockets of the receptors. This method is usually referred as ligand based drug design. The key advantages of database searching is that it saves synthetic effort to obtain new lead molecules. The very first advantages of ligand based or database searching is that our efforts to synthesize our drug molecules can be reduced if we use the database searching. Database is present online data. There is a big chemical space. Million and trillion organic molecules are present in the market. Furthermore, a lot of drugs which are tested, which are going to be used by the human, are present in the market. This is called database searching. From these drugs, we do hit to trial, we do random screening of the compound to obtain new chemical entity. But chemical entity is already present, but it has different biological action. Means it's nowadays there a news comes that coronavirus can be disease can be treated with dexamethasone. This is a steroid. This is already being used in number of cases, like in asthma, in skin diseases. People are frequently using the dexamethasone. But in Britannia, in British, the scientists have discovered that dexamethasone is also used to for the rapid recovery of coronavirus patient. There is a 90% effect of this dexamethasone on the patient. They have tried, but these are the trials. Still research is going on because this is the new disease. When the new disease comes in the market, now this is the very good example of structure based drug design. That first of all, what the scientists do, that they have discovered the structure of the coronavirus. They do homology modeling, they did genome sequencing. When the genome sequence come in the market, now it is easy. Usually cytokine protease inhibitors are used as antiviral agents. Cytokinase, proteases, these are used as these are used to enhance the viral load in the body. Cytokinase, proteases in the body are present, which invite the virus to develop a big viral load. That is why when you are going to inhibit the cytokinase protease inhibitors, then the viral load can be reduced. That is why ligand based drug design, the first category is about finding ligands for a given receptor, which is usually referred as database searching. In this case, a large number of potential ligand molecules are screened to find those fitting the binding pocket of the receptor. This method usually referred as ligand based drug design. The key advantage of database is that it saves synthetic effort to obtain new lead molecules. Next slide, please. Receptor based drug design, another category of structure based drug design method, is about building ligands, which is usually referred as receptor based drug design, because our ligands are usually receptors. They receive lone pair of electron, they receive hydrogen bond, they have the ability to accept lone pair of electron, they have the ability to accept hydrogen proton acceptors. That is why these are called the receptor based drug design. In this case, ligand molecules are built up within the constraints of the binding pockets by assembling small pieces in a stepwise manner. These pieces can be either individual atoms or molecular fragments. The key advantage of such a method is that novel structures not contained in any database can be suggested. The main advantages of such method that the novel structures not contained in any database can be suggested. We can suggest a novel structure that not contained in any database can be suggested. This is a receptor based drug. This is the flow chart of two strategies of structure based drug design. You can see that is the pharmacophore identification. In the next slide, you can see that pharmacophore identification, then there is pharmacophore modification, then fit for receptor, then potential drug. These are the flow chart. Again, active site identification, ligand fragment growing, then fit for receptor, complete growing, then potential drug. If there is fit for receptor, if it's not, then change the fragment. There is need to change the fragment. This is the flow chart for two strategies of structure based drug design. Structure based drug design: natural ligand screening, molecular biology or protein chemistry, 3D structure determination of target and target ligand complex modeling, drug design cycle, structure analysis and compound design, biological testing, synthesis of new compound if promising, then preclinical studies. I have discussed a lot about the clinical trials and preclinical trials. Structure based drug design, again ligand database for target proteins. Next slide please. Ligand database, target proteins, molecular docking, ligand docking into protein active site, pharmacokinetics and pharmacodynamics optimization. These are used as structure based design. If you have any question, you can ask me now, but you can put me your question through your WhatsApp, through Google Classroom, whatsoever you can ask me. We will discuss a lot about the structure based. Next, on the base of structure, we discuss about the docking, how docking help in designing the drug molecules. Then I will give you some assignments. The assignment will be uploaded on the Google Classroom. Okay, Allah Hafiz.